Hearing and vestibular sensation. Audition, or hearing, is important to humans and to other animals for many different interactions. It enables an organism to detect and receive information about danger, such as an approaching predator, and to participate in communal exchanges like those concerning territories or mating. On the other hand, although it is physically linked to the auditory system, the vestibular system is not involved in hearing. Instead, an animal's vestibular system detects its own movement, both linear and angular acceleration and deceleration, and balance. Sound. Auditory stimuli are sound waves, which are mechanical, pressure waves that move through a medium, such as air or water. There are no sound waves in a vacuum since there are no air molecules to move in waves. The speed of sound waves differs based on altitude, temperature, and medium, but at sea level and a temperature of 20 C, 68 F, sound waves travel in the air at about 343 meters per second. As is true for all waves, there are four main characteristics of a sound wave: frequency, wavelength, period, and amplitude. Frequency is the number of waves per unit of time, and in sound is heard as pitch. High frequency is greater than or equal to 15.000 Hz. Sounds are higher pitched, short wavelength, than low frequency. Long wavelengths is less than or equal to 100 Hz. Sounds. Frequency is measured in cycles per second, and for sound, the most commonly used unit is Hz, Hz, or cycles per second. Most humans can perceive sounds with frequencies between 30 and 20,000 Hz. Women are typically better at hearing high frequencies, but everyone's ability to hear high frequencies decreases with age. Dogs detect up to about 40,000 Hz. Cats, 60,000 Hz. Bats, 100,000 Hz. And dolphins 150,000 Hz. And American shad, Alosa sapidissima, a fish, can hear 180,000 Hz. Those frequencies above the human range are called ultrasound. Amplitude, or the dimension of a wave from peak to trough, in sound is heard as volume and is illustrated in Figure 1. The sound waves of louder sounds have greater amplitude than those of softer sounds. For sound, volume is measured in decibels, dB. The softest sound that a human can hear is the 0 point. Humans speak normally at 60 decibels. Reception of sound. In mammals, sound waves are collected by the external, cartilaginous part of the ear called the pinna, then travel through the auditory canal and cause vibration of the thin diaphragm called the tympanum, or eardrum, the innermost part of the outer ear, illustrated in Figure 2. Interior to the tympanum is the middle ear. The middle ear holds three small bones called the ossicles, which transfer energy from the moving tympanum to the inner ear. The three ossicles are the malleus, also known as the hammer, the incus, the anvil, and stapes, the stirrup. The aptly named stapes looks very much like a stirrup. The three ossicles are unique to mammals, and each plays a role in hearing. The malleus attaches at 3 points to the anterior surface of the tympanic membrane. The incus attaches the malleus to the stapes. In humans, the stapes is not long enough to reach the tympanum. If we did not have the malleus and the incus, then the vibrations of the tympanum would never reach the inner ear. These bones also function to collect force and amplify sounds. The ear ossicles are homologous to bones in a fish mouth. The bones that support gills in fish are thought to be adapted for use in the vertebrate ear over evolutionary time. Many animals, frogs, reptiles, and birds, for example, use the stapes of the middle ear to transmit vibrations to the middle ear. Transduction of sound. Vibrating objects, such as vocal cords, create sound waves or pressure waves in the air. When these pressure waves reach the ear, The ear transduces this mechanical stimulus, pressure wave, into a nerve impulse, electrical signal, that the brain perceives as sound. The pressure waves strike the tympanum, causing it to vibrate. The mechanical energy from the moving tympanum transmits the vibrations to the three bones of the middle ear. The stapes transmits the vibrations to a thin diaphragm called the oval window, which is the outermost structure of the inner ear. The structures of the inner ear are found in the labyrinth, a bony, hollow structure that is the most interior portion of the ear. Here, the energy from the sound wave is transferred from the stapes through the flexible oval window and to the fluid of the cochlea. The vibrations of the oval window create pressure waves in the fluid. Perilymph. Inside the cochlea. The cochlea is a whorled structure, like the shell of a snail, and it contains receptors for transduction of the mechanical wave into an electrical signal. As illustrated in Figure 3, inside the cochlea, the basilar membrane is a mechanical analyzer that runs the length of the cochlea, curling toward the cochlea's center. The mechanical properties of the basilar membrane change along its length, such that it is thicker, tauter, and narrower at the outside of the whorl, where the cochlea is largest, and thinner, floppier, and broader toward the apex, or center, of the whorl, where the cochlea is smallest. Different regions of the basilar membrane vibrate according to the frequency of the sound wave conducted through the fluid in the cochlea. For these reasons, the fluid-filled cochlea detects different wave frequencies, pitches, at different regions of the membrane. When the sound waves in the cochlear fluid contact the basilar membrane, it flexes back and forth in a wave-like fashion. Above the basilar membrane is the tectorial membrane. The site of transduction is in the organ of Corti, spiral organ. It is composed of hair cells held in place above the basilar membrane like flowers projecting up from soil, with their exposed short, hair-like, stereocilia, contacting or embedded in the tectorial membrane above them. The inner hair cells are the primary auditory receptors and exist in a single row, numbering approximately 3,500. The stereocilia from inner hair cells extend into small dimples on the tectorial membrane's lower surface. The outer hair cells are arranged in 3 or 4 rows. They number approximately 12,000, and they function to fine-tune incoming sound waves. The longer stereocilia that project from the outer hair cells actually attach to the tectorial membrane. All of the stereocilia are mechanoreceptors, and when bent by vibrations they respond by opening a gated ion channel, referred to. As a result, the hair cell membrane is depolarized, and a signal is transmitted to the cochlear nerve. Intensity, volume of sound is determined by how many hair cells at a particular location are stimulated. The hair cells are arranged on the basilar membrane in an orderly way. The basilar membrane vibrates in different regions, according to the frequency of the sound waves impinging on it. Likewise, the hair cells that lay above it are most sensitive to a specific frequency of sound waves. Hair cells can respond to a small range of similar frequencies, but they require stimulation of greater intensity to fire at frequencies outside of their optimal range. The difference in response frequency between adjacent inner hair cells is about 0.2%. Compare that to adjacent piano strings, which are about 6% different. Place theory, which is the model for how biologists think pitch detection works in the human ear, states that high-frequency sounds selectively vibrate the basilar membrane of the inner ear near the entrance port, the oval window. Lower frequencies travel farther along the membrane before causing appreciable excitation of the membrane. The basic pitch determining mechanism is based on the location along the membrane where the hair cells are stimulated. The place theory is the first step toward an understanding of pitch perception. Considering the extreme pitch sensitivity of the human ear, it is thought that there must be some auditory sharpening mechanism to enhance the pitch resolution. When sound waves produce fluid waves inside the cochlea, the basilar membrane flexes, bending the stereocilia that attach to the tectorial membrane. Their bending results in action potentials in the hair cells, and auditory information travels along the neural endings of the bipolar neurons of the hair cells. Collectively, the auditory nerve, to the brain. When the hairs bend, they release an excitatory neurotransmitter at a synapse with a sensory neuron, which then conducts action potentials to the central nervous system. The cochlear branch of the vestibulocochlear cranial nerve sends information on hearing. The auditory system is very refined, and there is some modulation or sharpening built in. The brain can send signals back to the cochlea, resulting in a change of length in the outer hair cells, sharpening or dampening the hair cell's response to certain frequencies. Higher processing. The inner hair cells are most important for conveying auditory information to the brain. About 90% of the afferent neurons carry information from inner hair cells, with each hair cell synapsing with 10 or so neurons. Outer hair cells connect only 10% of the afferent neurons, and each afferent neuron innervates many hair cells. The afferent bipolar neurons that convey auditory information travel from the cochlea to the medulla, through the pons and midbrain in the brainstem, finally reaching the primary auditory cortex in the temporal lobe. Vestibular information. The stimuli associated with the vestibular system are linear acceleration, gravity, and angular acceleration and deceleration. Gravity, acceleration, and deceleration are detected by evaluating the inertia on receptive cells in the vestibular system. Gravity is detected through head position. Angular acceleration and deceleration are expressed through turning or tilting of the head. The vestibular system has some similarities with the auditory system. It utilizes hair cells just like the auditory system, but it excites them in different ways. There are 5 vestibular receptor organs in the inner ear: the utricle, the saccule, and 3 semicircular canals. Together, they make up what's known as the vestibular labyrinth that is shown in Figure 4. The utricle and saccule respond to acceleration in a straight line, such as gravity. The roughly 30,000 hair cells in the utricle and 16,000 hair cells in the saccule lie below a gelatinous layer, with their stereocilia projecting into the gelatin. Embedded in this gelatin are calcium carbonate crystals like tiny rocks. When the head is tilted, the crystals continue to be pulled straight down by gravity, but the new angle of the head causes the gelatin to shift, thereby bending the stereocilia. The bending of the stereocilia stimulates the neurons, and they signal to the brain that the head is tilted, allowing the maintenance of balance. It is the vestibular branch of the vestibulocochlear cranial nerve that deals with balance. The fluid-filled, semicircular canals are tubular loops set at oblique angles. They are arranged in 3 spatial planes. The base of each canal has a swelling that contains a cluster of hair cells. The hairs project into a gelatinous cap called the cupula and monitor angular acceleration and deceleration from rotation. They would be stimulated by driving your car around a corner, turning your head, or falling forward. One canal lies horizontally, while the other 2 lie at about 45-degree angles to the horizontal axis. As illustrated in Figure 4, when the brain processes input from all 3 canals together, it can detect angular acceleration or deceleration in 3 dimensions. When the head turns, the fluid in the canals shifts, thereby bending stereocilia and sending signals to the brain. Upon cessation, accelerating or decelerating or just moving, the movement of the fluid within the canals slows or stops. For example, imagine holding a glass of water. When moving forward, water may splash backwards onto the hand. And when motion has stopped, water may splash forward onto the fingers. While in motion, the water settles in the glass and does not splash. Note that the canals are not sensitive to velocity itself, but to changes in velocity. So moving forward at 60 miles per hour with your eyes closed would not give the sensation of movement. But suddenly accelerating or braking would stimulate the receptors. Higher processing. Hair cells from the utricle, saccule, and semicircular canals also communicate through bipolar neurons to the cochlear nucleus in the medulla. Cochlear neurons send descending projections to the spinal cord and ascending projections to the pons, thalamus, and cerebellum. Connections to the cerebellum are important for coordinated movements. There are also projections to the temporal cortex, which account for feelings of dizziness. Projections to autonomic nervous system areas in the brainstem, which account for motion sickness. And projections to the primary somatosensory cortex, which monitors subjective measurements of the external world and self-movement. People with lesions in the vestibular area of the somatosensory cortex see vertical objects in the world as being tilted. Finally, the vestibular signals project to certain optic muscles to coordinate eye and head movements. Section summary. Audition is important for territory defense, predation, predator defense, and communal exchanges. The vestibular system, which is not auditory, detects linear acceleration and angular acceleration and deceleration. Both the auditory system and vestibular system use hair cells as their receptors. Auditory stimuli are sound waves. The sound wave energy reaches the outer ear, pinna, canal, tympanum, and vibrations of the tympanum send the energy to the middle ear. The middle ear bones shift and the stapes transfers mechanical energy to the oval window of the fluid-filled inner ear cochlea. Once in the cochlea, the energy causes the basilar membrane to flex, thereby bending the stereocilia on receptor hair cells. This activates the receptors, which send their auditory neural signals to the brain. The vestibular system has 5 parts that work together to provide the sense of direction, thus helping to maintain balance. The utricle and saccule measure head orientation. Their calcium carbonate crystals shift when the head is tilted, thereby activating hair cells. The semicircular canals work similarly, such that when the head is turned, the fluid in the canals bends stereocilia on hair cells. The vestibular hair cells also send signals to the thalamus and to somatosensory cortex, but also to the cerebellum, the structure above the brainstem that plays a large role in timing and coordination of movement. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.